Now, the most important thing, and I think it's a unique example of successful genomic medicine, was a treatment. I would say the cure of these people. Again, what you see here on the left is the potassium channel is permanently open. You don't secrete any insulin. First day of your life or first week of your life, you need insulin, exogenous insulin, and usually the result is not perfectly good. Now, a logical thing to do, but it was not so easy to do it because people and governmental organization was frightened to give sulfonylureas to newborn, was to switch to sulfonylureas. And in 90% of the cases, it works. You close the channel, and what you have is a cure of diabetes. That means that in few weeks, you can stop insulin, and these children have a normal fasting glucose, normal postprandial glucose. So they are normal if they take sulfonylureas. And that is a kind of miracle. Of course, the number of subjects is small, but it's a real progress. Now, so what did the genetic of diabetes bring to diabetes research? I think that as a basic researcher, first I would say that it brought some breakthrough about key beta cell development genes and about the pathway controlling insulin secretion in response to glucose. You probably say, okay, that's fine, but that's nothing to do with the discussion of today. And if you say that, I'm not sure you're totally right, because for two reasons. First of all, because if you see here, that is a beta cell, and here that the effect of the different genes that we and others found to be responsible for monogenic diabetes. And compared to what have been said before by my colleague from Eli Lilly, it's absolutely clear that it recapitulates what we know in type 2 diabetes. That means that monogenic diabetes is kind of summary of all the defects that you have in type 2 diabetes in general, and a good model to analyze potential new compound and treatment for type 2 diabetes, and possibly for type 1 as well. And the second very important thing is progressing a genetic of diabetes. You progress in the understanding of the development of major pancreatic cells from progenitor cell stem cell to something that can really mimic beta cells. So we have heard yesterday Professor Evans speaking about stem cell biology and future, and I think that this kind of genetic discovery can really help to develop, to engineer new beta cell for diabetes treatment. And Dr. Doug Melton published in Nature about 1 month ago the development of a new cell secreting insulin, and what he did is only to put some to express some gene that we and others have found to be responsible for diabetes in this progenitor cell, and he succeeded in making a cell that secretes insulin in response to glucose. Now, if we can really summarize what I think genetics of diabetes has brought so far, even if it's in general that was in a rare form of diabetes, it improved disease classification, it brought some individualized treatment, and possibly it will bring some new drug for diabetes, which I think is not too bad compared to the relatively small investment in the genetic of diabetes and relatively small number of groups that work on that. So I think academia have done not too badly for the translational research in diabetes working monogenic. Now, what about 95% of the patients who have a regular form of type 2 diabetes? So the situation is completely different in this case. It's a complex trait. The environment is crucially important. And as you see here, I like this quotation from my colleague from London, Steve Humphries: "Nature loads the gun, but nurture pulls the trigger," which I think is it's a good way to consider what is a multifactorial disease. What does it mean to have a polygenic disease? That means that individually, each of the genes, say gene 13, has a very small, modest role, and probably no role at all independently at the individual level. So to really increase the risk of diabetes, it should be some kind of interaction between different genes. It's a genetic background, not only one gene, plus the effect of the environment. What is the consequence of that? The consequence of that is that if you work gene by gene, you probably have no chance to really understand the picture. It's say like take a puzzle, you have one piece and say, oh, I see that, now you see nothing. So if you want to see everything, you need to have a global image of diabetes, which means that you need to have a whole genome approach of diabetes background if you want to have a chance to understand how it works. And that was a dream of about 5–10 years ago to do this kind of thing. I didn't prepare a slide on that, I'm sorry, because I expected Dr. what is his name, Carrie Stephenson, but he's probably blocked somewhere in Iceland. So what I want to say here is it has been possible because of three things. First of all, there was the sequencing of the human genome. And secondly, there was the HapMap, and the HapMap is funded by the Wellcome Trust and different other organizations. Is a kind of map of all the genetic differences that we have, and there's now about 4 million of these polymorphism differentially one individual to another that has been mapped by the HapMap consortium. The third thing is are two Californian biotech companies, and I have to say that I admire what they have done because and I admire the system of funding in America of these companies that failed 2 or 3 times and have been refunded again before they succeeded in developing microarrays. And this microarray is a real revolution for us because you can analyze up to 1 million of these polymorphism in one experiment for one individual for today about $200 or $300 for each individual. So that really made possible to analyze that, saying that the theory was that frequent polymorphism might play an important role in the genetic background of frequent diseases, and that is only partially true, but it's partially true. So you have the human genome, you have the map of the different, you have the microarray, and you have some funding, and you have the population. And then what you can do is you can do the experiment. And the question, does it work? And yes, it works. And that we are very lucky in my group, as our chairman said, that because we have published the first genome-wide association, not only in diabetes, but also in complex trait, using this new generation of microarrays. It was in February in 2007. And I think it was considered to be an important breakthrough by science, and the people in science were nice enough to really refer to our paper in December last year. And I think it was important not only because we found genes, but also because for the first time in genetics, the others also found genes. So Decode from Iceland found the same gene, the WTCCC, the Broad Institute, and NIH found the same gene that us, so replicated our result. And in addition, they found additional genes. And today, we have about, even now it's old-fashioned, this slide, there's probably 15 to 20 genes that for sure we know they contribute to the genetic background of type 2 diabetes. And for the first time for years, geneticists agree with each other what are the genes for type 2 diabetes. And believe me, that is a major achievement to have 2 geneticists saying the same thing. That is an achievement to have 2 physicians saying the same thing about a patient.